children, and welcome back. Today we are going to do the lesson number 10, which is Microbes in Human Welfare. Right? Now, not only these microbes cause diseases in human beings and other living organisms, they are also useful to us in many ways. From time immemorial, in the households, they have been using yeast in making bread, isn't it? Right. And you also will learn in this chapter how microbes, especially yeast and many kinds of bacteria, are used to produce many kinds of acids, many household products, enzymes, okay, and how it is used as biocontrol agent as well as biofertilizers. Okay. Do you know who converts milk into curd? Yes, yeah, it is lactobacillus or LAB, lab. It is called shortly pronounced as lab. If you add little bit of curd to the milk, okay, this little bit of curd contains thousands of lab or lactic acid bacteria which will convert or which will digest the protein and coagulate the milk, okay, and also it is used to synthesize vitamin B12 in our digestive system. Clear? Right, it not only converts milk into curd, it also produces vitamin B12. Okay, now this a little bit of curd we are adding to the milk now, which is called as inoculum or starter. Starter. It contains thousands of bacteria, lab bacteria. Okay, and which are the other common uses of microbes in the household, getting household products. Another microbe, yeast, which is a fungus, which is a commonly used microbe, which is used while making dough for idli, doshas, and bread and all in bakeries, in baking cakes and biscuits and all. The puffed up condition of these dosha and idli and bread is because of the large amount of CO2, carbon dioxide, produced by the action of this yeast on the food material. Okay, this process is called fermentation. Clear? And toddy. Toddy is also fermented by using by yeast, right? Microbes are also used to ferment fish, soya beans, and bamboo shoots to produce cheese. Okay, and the Swiss cheese, one of the cheese variety, has large holes on it because of the production of CO2 by the bacterium. It's caused by a bacterium called as Propionibacterium shermani. What is the name, children? Propionibacterium shermani. So this bacteria is a result of the large holes on the Swiss cheese. Okay, and Roquefort cheese is ripened by a particular type of fungus grown on it, which is called as Penicillium roquefort. Okay, Roquefort cheese, it gives a particular flavor or taste to that cheese because of this fungus is grown on this Roquefort cheese. Okay, right. Microbes like bacteria and fungi are also used to make beverages, many kinds of beverages. Okay, beverages are made in large fermenters, large tanks or vessels called as fermenters. Okay, right. Here also the same yeast which is used in the bakeries. Oh, it is called as baker's yeast. When it is used in bakeries, bread and making cakes and bread and all, it is called as baker's yeast. Okay, when it is used in the breweries to make beverages and all, it is called as brewer's yeast. Okay, right. And it is used to produce many kinds of beverages like wine, rum, whiskey, and all those things, right? Okay, and wine and beer are produced without distillation. After fermenting, without distillation, it can be used, wine and beer. Whereas the other ones, whiskey and brandy and rum, are produced with the distillation. Clear, children? So the same brewer's yeast, which is, what's the name of the yeast, children? What is the scientific name of yeast? Saccharomyces cerevisiae. Saccharomyces cerevisiae is the name of the yeast, the common yeast, which is used both in bakeries and breweries. Okay, right. And also antibiotics. Many kinds of antibiotics are obtained from bacteria and fungi. What do you mean by antibiotics, children? What are antibiotics? The name itself says antibiotics, against life. Okay, so here antibiotics are obtained from the fungus and bacteria especially, and it is used to treat the infections caused by bacteria and fungus. Okay, and do you know the first antibiotic obtained? Who is the person? Yes, it was Alexander Fleming who discovered penicillin. Now, how did he discover? Do you know that? Now, Alexander Fleming was working on Staphylococcus bacteria. Staphylococcus bacteria. And he had, after doing his experiments, he had left his petri dishes where he used to grow this bacteria unwashed. Okay, and he saw that some kind of mold, that's a fungus, was growing in this bacterial culture. And at the place where the mold was growing, the bacteria were not present. The staphylococcus were not present. Okay, and it was due to the secretion or synthesis of a particular kind of substance by the fungus. The fungus was Penicillium notatum. Okay, and the substance obtained, the secretion obtained by this from this species was called as penicillium. Right, so this was the very first antibiotic obtained, and it was used to treat various kinds of diseases, which was used in, which it was also used to treat soldiers, wounded soldiers in the World War II and all. Okay, right. So it was used to treat plague, whooping cough, diphtheria, leprosy, and various other kinds of diseases. Okay, and it was Ernest Jane and Howard Florey who established its full potential as an effective antibiotic. Okay, and for that they were awarded the Nobel Prize in 1945. Clear? And next you will see how microbes are used to produce many kinds of acids, enzymes, and bioactive molecules. Right? So what are the acids which are obtained from the different fungus and bacterium? Citric acid obtained from Aspergillus niger. It's a fungus, right? Acetic acid from Acetobacter aceti. So only this Aspergillus niger is a fungus, all the others are a bacterium. Okay, acetic acid from Acetobacter aceti. Butyric acid from Clostridium butylicum. Clostridium butylicum. And lactic acid from Lactobacillus acidophilus actually. Okay, so which are the acids, children? Citric acid, butyric acid, acetic acid, and lactic acid. Okay, and you already know that the yeast, which is Saccharomyces cerevisiae, is commonly used to produce various kinds of alcohols, especially ethanol, which is used in industries. Okay, right. And the various enzymes are lipases. What is lipase, children? Where have you heard lipase? Yeah, it is a fat digesting enzyme. So it is used, these lipases are used in the detergent formulations in removing oily stains. Okay, lipases, fat digesting enzymes. So it is used in detergents to remove oily stains. Children, when we make juices at home, it is more concentrated. But what we get from shops, the bottled ones, are more transparent. Isn't it? Right. How do you get that? How does it happen? It is because of the enzymes pectinases and proteases. What are they? Pectinase and proteases. They are used to clarify bottled juices. They are used to clarify, make it a little more transparent it becomes, right? Bottled juices, to clarify bottled juices. And streptokinase from streptococcus. It is obtained from streptococcus. It is used as a clot buster. What is a clot buster? It is used to remove clot from blood vessels in patients, which may cause heart attack in those patients, right? So to remove clots from blood vessels. So it is called as a clot buster. Okay, right. And okay, now again recall what are the three enzymes we are learning in this chapter. It is, yes, lipases used in the detergent formulations to remove oily stains. Pectinases and proteases used to clarify bottled juices, and streptokinase from streptococcus, right? And it is used as clot busters. Okay. Next we'll see some bioactive molecules obtained from microbes, right? Now, cyclosporin A, which is produced from a fungus called Trichoderma polysporum. Which is the bioactive molecule, children? Cyclosporin A, obtained from Trichoderma polysporum, which is a fungus, right? Okay, and it is used as an immunosuppressant agent. Children, those patients getting a transplant from other persons, right? Their immune systems may overreact and reject the organ, thinking that it is not there, its bodies, right? And it will reject, and the patient may die. The operation may be success, but the patient dies, and the organ may not function properly in that patient, right? So for that purpose, the suppressing the immune system, not to reject that organ, transplanted organ. Okay, so the cyclosporin A is a immunosuppressant agent. Okay, and statin is obtained from another fungus called as Monascus purpurus. Only two bioactive molecules studying, children. You please remember both of them, right? For statins are obtained by Monascus purpurus, and it is used as a blood cholesterol lowering agent. What is the purpose? It helps to reduce blood cholesterol level. Okay. Next we'll see how microbes are used in the treatment of sewage. Now what is sewage, children? Sewage is the dirty water or the waste water which are produced from the different households, different industries, everything, right? So most of the industries, they are located on the banks of the rivers or any water bodies, and they just release these waste from the into their water bodies. Okay, but actually in most cases it is treated first. This is treated in sewage treatment plant, and then only it is released into the water bodies. Right. It includes two kinds of treatment, which is the primary treatment and the secondary treatment. Okay. In the primary treatment, what is done is mainly the solid particles or the effluent is removed by the process of filtration. Mostly solid particles and all those will be there. Now, all those are removed by sedimentation, and also the top flowing items and all will be removed by filtration method, which includes the primary treatment. The settled solids form the primary sludge, called the primary sludge, and the supernatant forms. The supernatant means topper, the liquid in which there is dissolved materials plus floating debris and all. No, that forms the primary effluent. So what are the two things in the waste, sewage waste? The primary sludge, which is the solid things, and the primary effluent. Now comes the secondary treatment, which is actually the biological treatment, where the aerobic and anaerobic bacteria and fungi act and reduce the different kinds of waste to a considerable level. Okay, so here the secondary treatment, the primary effluent, the top layer, that is passed into large tanks, which is called as aeration tank, because oxygen is pumped into that, and it is also kept agitated means kept shaking, that the entire vessel is kept shaking, or more, the water is allowed to constantly move. Okay, so which is called, this tank is the aeration tank. Okay, the effluent is passed to an aeration tank and it's constantly agitated. But this will grow continuously. Oxygen is also pumped. Okay, there is bacteria present, aerobic bacteria as well as anaerobic bacteria is present, already present. Okay, but this allows the vigorous growth of useful and aerobic bacteria, and also it also combines with the fungus, some kind of fungus growing in that to form a mesh like that is the top layer of the water will show some filament like structures or mesh like structures called as together, that is actually bacteria plus fungal growth, which is they grow in large amount, which is called as flocks. What are flocks, children? Bacteria with fungal filaments to form mesh-like structures on top of the water bodies, which are called as flocks. Okay, these microbes consume the organic matter in the effluent. This reduces the BOD. What is BOD? Biological oxygen demand of the effluent. They use the oxygen. These bacteria, these fungi, they start growing and they produce a large amount of their numbers. They also use oxygen. They also consume the nutrient present in the water bodies, and they reduce biological oxygen demand to considerable amount. Okay, now biological oxygen demand, or it is the amount of oxygen consumed by bacteria to reduce the organic matter in 1 liter of water. It is the amount of oxygen consumed by bacteria to reduce the organic matter in 1 liter of water. Okay, right. So it is actually BOD is a measure of organic matter present in the water. The greater the BOD, the more is its polluting potential. Clear, children? Now, after the BOD is considerably reduced, now this effluent with the flocks is passed into another tank, which is called as settling tank. It is passed through to a settling tank where the bacterial flocks and this sediment is called as activated sludge. What is that? It is called as activated sludge. What is done, children? The flocks are there now. Flocks is now passed to a settling tank. Now where the bacterial flocks are sedimented. They are sedimented, right? And this sediment is called as activated sludge because there is bacteria and fungi in it. That's why it is called as activated sludge. Okay. Now little bit of this activated sludge is passed back into the aeration tank. Again, little as a starter, as an inoculum, because it contains lot of bacteria and fungi, right? So it is passed back into the aeration tank. Okay. Now the remaining sludge, that is along with the flocks, bacteria plus fungi, is pumped into large tanks which is now called as anaerobic sludge digesters. Anaerobic sludge digesters, which will digest. Now the anaerobic bacteria come into action. Anaerobic bacteria, right? The anaerobic bacteria digest even the flocks also, and the remaining nutrients present, the organic matter of the waste or the water body or the sewage is digested by the anaerobic bacteria. Okay, and now also this also produces, during their action they also produce gases like carbon dioxide, methane, hydrogen sulfide, all these things, right? And this methane it can be used as a biogas. So it can be pumped through pipelines and it can be used as biogas, as a fuel, right? And now the water body, the sewage is considerably, the waste in the sewage is considerably reduced, and now it is ready to be released back into the water bodies. Clear?